done as we.
Hi, everyone. So my name is Sham Nair. I'm currently the Science Advisor 7 to 12 at the Department of Education. So I'm going to talk to you a little bit about Module 3, particularly the statistical testing parts of that module. So that part can be concerning for some students. But the thing is, it's actually quite a straightforward set of ideas that you need to be comfortable with. And I'll try and go through most of that today. Okay, I'm going to share my screen. So just bear with me. Okay, is that displaying a Word document?
Yes.
Okay, cool. Now I'm going to initially skip that screen, but I want to, for those of you who's just joining us, there are two files in the Google Drive that I've uploaded. There are these two. There's a PDF file, okay, this LMC SciX 2021 Study Day, Module 3 Statistics, and then the accompanying Excel file. So I'm going to be referring to both of these files in this presentation. So if you want to follow along, refer to both of those files. So, okay, and I'm going to jump between screens as well as we're doing this. So do bear with me as I come to grapple with technology. As you know, for the department schools, we've had a bit of a technical issue with the cyber attack and so on. And so things are not going as smoothly as we'd like. And also for the purposes of bandwidth and so on, I'm going to actually turn off my video. Okay, so if you'd like to, as I said, refer to the PDF file and the Excel file. So I'm going to now switch screens to the, this is the PDF file. I've just got it open in Word. So that's me, and that's my email address. Okay, and this is the document that I put together for today's session. Okay, the first part of this document just basically lists the syllabus statements or the syllabus descriptions. But I'd like to start here. So why why statistical thinking? So one of the aims of science extension is to try and get science extension students to think like a scientist. In other words, to think critically, logically, and using evidence wherever possible to support their statements. So if you look at a figure like this one here, actually, let me just close the navigation window pane for now. So if you look at this sort of a figure, you see an example of a spurious correlation. So here we have in red the mozzarella cheese consumption data for the US. And the number of doctorates awarded in civil engineering shown as the black line. Now, we all know that, this is obviously just a spurious correlation because it just occurs by chance. There's no causation or cause and effect relationship between these two phenomena. But it can become significant. So if you look at this graph here, this is actually from a publication that this was a while ago, that looked at the usage of, or the uptake of the MMR vaccine. So that's measles, rubella, and so on. So that is shown as the red line. So on the horizontal axis, you've got birth year, and then on the vertical axis on the right is the MMR, vaccine coverage. So this is the rate of vaccination shown in red. And in blue, we have the incidence of autism. Now, this was actually in a published paper. And what the author suggested was that there is a correlation between the use of this vaccine, and it may actually cause autism. Now, when you look at this graph, if you look at the two axes, you'll notice that one starts at 0. The vertical axis on the left starts at 0. The other one does not. In fact, actually, there's no correlation between the two. Okay, but this was where a graph was concocted to give the appearance that there is a relationship between these two variables. Now, it had serious consequences. In the UK, the rate of measles went up 200% in a matter of, I think it was about a year or so after the publication of this paper. Now, this paper has now been discredited. It's now been taken down, but it can have very, very serious ramifications. Now, you as science extension students would be asked to reflect on the validity of information like this. Here's another example. This is an interesting one. So this is a research that was done by a group in China, and they're looking at developing computer algorithms or software which can identify potential criminal behavior. So what they did was they had this software that they developed, and then they used something in that we call in research circles testing sets. So in testing sets and training sets, actually, there are two terms that I use. What they do is they feed information into the algorithm. The algorithm processes that and then is able to predict certain outcomes. So what they did was obviously showed a bunch of photos of, and I think I've got it on the next page. Oh, here, a bunch of photos of people with criminal records, which is on the top, and then a bunch of people with who didn't have criminal records. And so they fed this sort of information into the algorithm, and then they said that they could predict, this particular software could predict criminal behavior with an accuracy of something close to 90% or something like that. Now, again, from a statistical perspective, that's rubbish. You can't do that, because often you find things like, if you look at the photos of people with a criminal record, and if you look at the paper, where did they get those photos from? Generally from police records. And so you can imagine people who are being apprehended for criminal activities are not going to be smiling into the camera, right? Compared to, photos of the general public. So there's already a bias in the way that the data that was used to feed into this algorithm. There's already an inherent bias. And that bias became amplified when they tested it, okay? Once again, as science extension students, you need to be able to think about the way data is collected, processed, and analyzed, okay? And that's going to be part and parcel of your exam. In your exam, you'll be given all sorts of data from different types of experiments. And you'll have to think about the way that the experiments were conducted. So understand the descriptions, look at the data, the way it's presented, and be able to read between the lines, so to speak. Okay, that's what scientists do. Okay, they don't take data at face value. They look at all the other metadata associated with all the data sets. And I'll tell you a little bit more about it as we go through this session. Okay, so those are just some introductory messages about the importance of statistical thinking in science. Okay, so one of the things we all need to know as far as the syllabus is concerned is the distinction between descriptive and inferential statistics. So statistics is broadly defined into these two groups, descriptive stats and inferential stats. Now, the names actually tell us what these different types or categories of statistical tests are used for. So descriptive statistics basically describes the properties of data. Inferential allows us to make inferences. Okay, so inferences means to draw conclusions, and we can actually extrapolate. So let's go back to descriptive data. We have two types, the ones most of us are familiar with, the measures of central tendency, so looking at things like means, medians, and so on. And then you have distributions, and you should know what the normal distribution is, and I'll come to that in just a tick. Okay, so all of this falls into the category of descriptive statistical tests. Then we have the inferential tests. And once again, for our syllabus, you need to know that inferential tests are used to either establish relationships, which means correlation, establish correlation, or where you compare different data sets. So that means comparing means or variances. So in a nutshell, that's what the module 3 is all about. So in terms of understanding how statistical tests are done, just know that we have descriptive and inferential. And I'll go through some of these tests in just a bit. Okay, so that's descriptive. The syllabus actually also requires you to be familiar with statistical software. And when I say statistical software, it means any tools that you would have used in your classrooms to do some statistical analysis. So Excel is the most popular one. It's not the best, but it is the most popular. And it requires an add-in, and I'll show you that in just a tick. So once it's properly installed and configured, under the data tab of Excel, you will see this data analysis thing come up. And that's the descriptive stats that we will be using. And in Excel, you also have a whole bunch of other tools, statistical tools, and they can be used for things like F-test and T-test and chi-square test and so on. As I said, Excel was not designed for statistical testing. So sometimes for a particular test, you might have to do it in 2 or 3 distinct steps. So for example, if you're going to use t-tests in Excel, you need to do an f-test first, followed by a t-test. So it can be a little clunky, but nevertheless, we are all familiar with spreadsheets and how to use Excel. Another one is a good online software that we can use is Vasastats. It's free. And once again, I'll give you an example of how we use that in just a tick, just in case you haven't used it in your classroom. Next, you need to know the distinctions between the different types of data. Okay. So data generally are divided into qualitative and quantitative. So the word quantitative, quant means to measure. That means you're going to be measuring some variable. Qualitative means that it's a categorical. Okay, you're looking at the quality of some variable. On the left, we've got categorical data which are further divided into nominal and ordinal. So nominal, the word nom in Latin or French means name, okay? And what that refers to is variables in no specific order or sequence. Okay, so if you look at eye color, so you might say brown, blue, green. There's no specific ordering. Okay, and blood groups can be A, B, and O. Ordinal data, again, it's a categorical or a qualitative data that actually has a ranking order. So if you look at, say, exam grades A, B, C, D, E, A is higher than B, which in turn is higher than C, and so on and so forth. So that's called ordinal. The word ordinal comes from order. Okay. On the other side, we've got the quantitative types. Now, quantitative data sets, they can be a little bit more complicated, but essentially we've got two groups: discrete, those that actually fall into specific groups. For example, the example I've got here is, if you think about sport, the number of wins and losses for a particular competition, okay, they can only be in whole numbers. So they're discrete quantitative variables. Continuous variables are those that have all the numbers in between, okay? It can be subdivided. So if you look at height measurements, a person can be 1.5, 1.51, 1.55, 1.58 meters tall, and so on and so forth, right? So the number of decimal points can increase or decrease as much or as little as we like. So that, and the reason why we need to know about the type of data that we are working with, that's because it's going to determine the type of statistical test that we can use. Okay. So most of that should be straightforward, but I want to actually delve a little bit more into this whole thing about qualitative and quantitative data. Sometimes the distinction is not clear. I'll give you an example here. Let's look at something like blood group distribution. Let's say you go to the Australian Bureau of Statistics, which has data sets on the proportions of Australians in the AB, and O blood groups. Now, think carefully about that data. Okay, blood groups themselves are qualitative because we've got A, B, and O. Okay. There's no specific rank order to the groups, so we say that they are nominal qualitative data. However, as soon as you start putting in numbers into that, say the proportions of Australians who are A blood group, proportions, or who might be B, etc., we actually change that variable from nominal qualitative to discrete quantitative. So we're still looking at the same variable, but determining whether it's qualitative or quantitative depends on what you're trying to do with that variable. Okay, and that's something to bear in mind. I've given a couple of other examples as well. So again, a simple example is exam grades. Okay, so at the end of year 11 in this IS course, students were awarded a grade A to E. Now, A to E is qualitative, but because there is a rank order to it, it's ordinal qualitative. Now, as soon as you put in the proportions of students who received A, B, C, D, and so on, it becomes discrete quantitative, right? So, and that's a key message that I'd like to send to you. When you're looking at a variable, look very carefully at what actually is being measured and reported. Okay, is it just the groupings, or is it looking at the measurements within those groups? Because that determines the type of data you're dealing with. So the third example there is you can look at it later. Okay, so now we've looked at what we mean by data, the data types, data quality, the measurements, and so on. Let's go take a step back and look at the two big groups of statistical tests, descriptive and inferential. I'm going to start off with descriptive because it's the easier of the two, and most of us would have looked at descriptive statistics both in maths and in this course as well. And obviously you might have done it in the other science courses. Okay, so descriptive stats. So and I want to bring your attention to this sentence here. Okay. If you take any repeated measurements, okay, it doesn't matter what it is, okay, it can be qualitative, quantitative, whatever, that's not important. One of the things about descriptive statistics is this thing called central tendency. That means given a set of measurements, okay, given a set of measurements, there's always a center to that data, to that data set. That center is actually quite important, and how we determine that center is also quite important. Okay, so what do we use generally? Well, the center is defined as mean or median, generally speaking. We also use mode, but mean and medians are the two common central tendency measures. In Excel, mean is called average and median is called median. So if you just type in the formula box equals average and you select your column of numbers, it will calculate the mean for you. And then we have two other measures here, variance and standard deviation. Okay, they are both used to indicate how far the data spreads from that center, from the mean or the median. Okay, and again, here are the Excel formulas that we use. One point, if you're working with large data sets, is it's often better to use median than means, because with large data sets, you find that the mean tends to bounce around a lot because of its sensitivity to outliers. Okay, so outliers in any data set are those extreme measurements, extreme numbers in those data sets. They tend to affect means more than medians. Okay, so it's just a point to note. It's not very important. The next thing, so we look at the central tendency. The next thing about descriptive statistics is something called the normal distribution. Okay, the normal distribution is kind of a funny term. Back in I think it was in the 18th century, when mathematicians were sort of calculating and measuring all sorts of natural phenomena, they noticed this distribution pattern. So numbers, so if you counted, for example, the lengths of all the leaves in a tree, they would tend to fall into this of a pattern, a distribution pattern. And so they called it normal. Technically, it's called a Gaussian distribution, but we've all stuck with normal distribution. So what are the main features? Well, you see a central hump in the middle that indicates the most commonly occurring measurements. There is in dotted line, the dotted line in the middle, which shows the mean or the median or the mode, whatever the central measure is. And the other thing is that the graph or the distribution is symmetrical around the center. These end bits are called the tails. Now the tails are asymptotes, okay, so they don't actually touch the horizontal axis. So that is a normal distribution. Now, why is it important? It's because of this. Every time there is a normal distribution, okay, we get these features. And it happens to be the same no matter what you're measuring, okay? Just to explain that to you using this figure, here's a normal distribution, okay? That's your mean, which is 0, okay? The dark blue represents 1 standard deviation. So that's 68% of all data points will lie there. And then 95%, so from here, the 2 sigma to minus 2 sigma, okay, this whole bit here, 95% lies within 2 standard deviations, and 99.7, remember, it never touches the asymptote, so it's 99.7 will lie within 3 standard deviations. This becomes important when we consider something called confidence interval later on. I'll come to that later on. We always use 95% as our rule of thumb, okay, the p-value and things like that later on. And that's because we want to know if our data lies within this part of the distribution, okay, between the 2 standard deviations from the center. All right. Now, there's another interesting thing about normal distributions. If you increase the number of measurements, you actually change the shape of the normal distribution. There's still normal distribution, so you still have the center, you still have the tails, and so on and so forth. What this figure is showing is that the blue line here shows when you only have 1 data set. So imagine you're doing an experiment just once, okay? That will have a distribution like that shown by the blue curve. If you repeated the experiment 3 times, so you have a total of 4 measurements, okay? You still have a normal distribution, but look at what's happened. It's kind of it's squeezed in towards the center, and the peak has gone higher, right? If you repeated your experiment 7 times, a total of 8 measurements, you see the same phenomena happening, okay? The curve has become taller and skinnier. But what this graph is showing is that as the number of samples increase, so in other words, the more you repeat some observation, the smaller your standard deviation is going to be. So if you look at the spread from the center, okay, so imagine this center here, which is set at 100 for this particular graph, okay, the distance from the center, okay, it gets narrower when you increase the number of samples, okay? So one of the things that you need to know as a science extension student is the way to improve the accuracy of your results. Okay, one of the ways of improving the accuracy of your findings is to increase sample size. Okay, and the reason for doing that is shown on this graph. Okay, now you can say that if you increase the sample size, you will decrease the standard deviation and the variance. Okay, both of those are decreased. Okay, so remember, in science extension, it's always important to show your reasoning skills. Now here in this figure, we've got the reason for why it's important to increase sample sizes in our investigations. So I've talked a little bit about that in the next section, so when we're talking about samples, okay? Always try and increase the number of samples that we will use for any studies. So if you're asked in the exam to design an investigation, make sure that you address that particular feature. So the number of times that you might repeat the observation or the measurements, and sample size. Okay, these are the two things that you need to address as a science extension student. So what do we mean by a sample? That means the group that you're actually studying. So in this figure, imagine that this is the population. Oh, by the way, in statistics, when we say population, it doesn't mean every person on the face of the earth. Population means all instances of whatever it is that we are studying. Okay, so it's used in a slightly different way to our everyday conversations. So population means, for example, if you're doing a research on high blood pressure, for example, then the population would be every person with that condition. Okay, your sample is always drawn from the population. Okay, so your sample is always smaller. And what we're trying to do in inferential statistics, remember the second category, in inferential statistics, we're analyzing samples and we are making inferences about the population. So, currently, with the COVID pandemic, we have all these companies developing various vaccines. And what do? They will study the effectiveness of the vaccine on small samples, okay, on a number of different samples, obviously, but a small sample relative to the population. But after obtaining data from the samples, they will then project that finding to the whole population. And that's what we mean by statistical inference. You do your study in a small sample and you project the findings to the whole population, because your sample is drawn from the population. Okay. All right. So I've given some points about sampling. Just read through this. They're quite straightforward. Okay. Now I want to briefly swap screens to the Excel spreadsheet. So you will have this Excel spreadsheet, as I said. So in this Excel spreadsheet, we've got a number of tabs at the bottom. No, I can't see the tab. I've just got to remove this. Okay, sorry about that. It automatically cut me out, muted me. I've given you this Excel spreadsheet, which contains a number of data sets. Okay, so each data set is represented in a separate worksheet. These, for all except this one here, which has exam marks, there are some activities relating to those data sets in the PDF file or the Word file. Okay, so I mentioned earlier that with Excel, okay, you should have this data analysis tab. So to access the data analysis tab, you would go into that data tab first, and then on the right, you have data analysis. This worksheet here shows the marks for a yearly exam. Okay, so it's a randomly generated data set. We can do descriptive statistics at the click of a button. Usually you'll get an error warning. And these are all the different statistical tools that's available in this particular package, okay, the data analysis package. There are actually more statistical tests in Excel, but this particular data analysis package only brings up the ones shown in this box. So let's say we want to do a descriptive statistics test on this data set. So we select that option, then you get a dialog box. The input range is from the first data point all the way to the last data point. Okay. I don't have labels, so I'll ignore that. Output range will be, I'm just going to click randomly anywhere on the worksheet. Now, what do I need? I need summary statistics. And let's say I want to know the confidence intervals for the mean. So I've selected those two options and click on OK, and it will produce a table. Now this table does a lot of the, as I said, it does a lot of the statistical analyses automatically. So we don't have to do this individually. You could actually go equals average of that data set to get the first row here, and then calculate standard errors, calculate median, etc., etc. You know, we don't want to do that when they can shortcut the whole process. Okay, so here in this result, what it's telling us is here's the mean, if that's what you're interested in. There is the median. Okay, mean and median are very close together, okay, which is actually a good thing. Here's your standard deviation, okay, and here you have a number of other measurements like range, what the maximum mark is, what's the minimum mark, and so on. And right at the bottom, you have something called confidence interval. So what does this tell us about the data set? It's telling us that the average mark for this group of students is 69. Okay, but there is a standard deviation of almost 19. So if we say, let's round this off, let's say that is 70. Okay, 70 plus, I can't insert symbols here, but basically it's a plus minus, okay, and the standard deviation, and let's just round it off for argument's sake to 19. Okay, so what it's telling us is that for one standard deviation, it'll be from 70 plus 19, okay, and 70 minus 19. So that's the range for one standard deviation. So that's what Excel allows us to do. You get basic descriptive statistics, the central measure, the standard deviation. You also get a whole bunch of other stuff like variance, okay, if you're interested in variance. And it tells us you can actually increase this list of tests. As science extensions, you just need to be familiar with using this type of data. I'll show you an example of where of a NESA sample exam question where you actually use the standard deviation data to argue the validity of the experiment. Okay, so here, this is Excel as a software tool. I will show you another one, that the Vasistats, in just a bit. So let me change, actually, well, I've got you there. So that particular data set, the assessment data, if you plot it, it will look okay. That median, oh, sorry, I'm trying to draw freehand, was 69. Okay, and we said we'll round it off to 70. And this then represents the standard deviation. So it's 89 at the higher end and 51 at the lower end. So this is one standard deviation from there to there. Okay, so make sure you're able to interpret those numbers. Okay, so once again, that is the mean or median at 70 for the exam marks data set. Okay, that's the median in the middle. And then we've got one standard deviation to the left and one standard deviation to the right. Let me go back to this. All right, so that's basically all that I wanted to say about descriptive stats. Now let's look at inferential stats. Now, as you, if you have questions, as Mr. Matchett said, do post them in the chat, and I'll come to those questions a bit later, okay, towards the end of this presentation. So you need to know, you will not be asked to actually do a statistical test in the HSC exam, because obviously you don't have the tools to do it. But you do need to know about the different types of tests, particularly the ones that are described in the syllabus, okay? You need to know what are the conditions in which particular tests can be used. Now, this flowchart might help you simplify that conversation. So let me just move this out of the way again. Okay, so let's start where, continue that conversation we had earlier. So data can be quantitative or qualitative. Okay. Qualitative data, essentially there's only one type of statistical test that's mentioned in the syllabus, and that's the chi-square test. Okay, so there are two types of chi-square tests, depending on the number of groups that you're working with. If it's one group of data, you use the goodness of fit. Two groups of data, you use the contingency. Really, there's not a lot of difference between the two. Okay. Now we come to this side on the left, the purple. We have the quantitative data. Once you have the quantitative data, there are, and you might remember me mentioning earlier on, there are two things we can do. One is to establish relationships, okay, the relationship between two variables, and that's only one test described in the syllabus, which is the Pearson correlation. With quantitative data, the other thing we can do is find the difference between the two data sets. If you use variance to measure the difference, then you would use the F test. If you're using the means, the average, then you would use the T test. Okay. So here, what this figure shows is, and let me just, what this test here shows is that, just here. It's an overview of the different types of statistical tests. Okay, you can use this chart as a decision flow. Okay, so starting at the top, we work our way down depending on what you're trying to accomplish in your analysis. I will show you once again how to use this flow chart a little later. Okay, but here once again are the key points. If you're working with qualitative data, there's only one test that you need to know of, chi-square test. If you're using quantitative data, then Pearson to determine the correlation coefficient or the relationship between two data sets. Or if you're comparing data sets, then you would use either the F test or T test, depending once again on what data you're working with. So this page is quite important, okay, because it gives you that overview of all the different types of tests. So now let's bring all of that together. What are the contexts of the questions that you might expect in the HSC exam? Why use these different statistical tools? The main reason, again, for this syllabus, if you do statistics at university, you'll learn about many other things. But for our syllabus here, what we're focusing on is hypothesis testing. So at the start of a research investigation, we have a hypothesis, okay? That's called a scientific hypothesis, okay? So your hypothesis might be, and I've got an example here, okay? So let's start here. Let's say someone is doing a study on the effects of physiotherapy on post-surgical patients. So here, the hypothesis states patients undergoing physiotherapy after shoulder surgery recover faster than those who do not undergo physiotherapy. Okay, so this is the scientific hypothesis. To do a statistical test, you then have to develop two other hypotheses, the null hypothesis and the alternate or the alternative hypothesis. Okay. Remember, the scientific hypothesis is the foundation. That's the thing that forms the basis of the research project. Okay. The statistical testing is done after the data has been collected. Okay, not before that. So understand, and what this page tells us is it provides us with a list of definitions. So a null hypothesis, and the word null means to nullify the scientific hypothesis, okay? So for example, if we were to say, going back to this one, the scientific hypothesis says patients undergoing physio will recover faster than those who do not. Then the null hypothesis basically says that that is not true. So we'd say that they do not recover faster. Okay, I hope that makes sense. So the null hypothesis simply negates the scientific hypothesis. It nullifies the scientific hypothesis. Once you've gotten your null hypothesis, you then have the alternative hypothesis. The alternative hypothesis is alternative to the null, okay? And so it is very similar to the scientific hypothesis. We have this word here, p-value. P-value stands for probability. Okay, that's what the P stands for. So how probable is it that the null hypothesis is true? Confidence interval. What is the confidence range that you're willing to accept for your study? Now, I mentioned earlier that we generally tend to use 95%. So that's 2 standard deviations, based on the normal code, okay? So 95% expressed as a decimal is 0.95, okay? So that's the confidence interval. The alpha value is the significant values, significance value, or the critical value. Okay, but they all mean the same thing. And this is basically the value outside of your confidence interval. Okay, so what I'm going to do is I'm going to go back to this and okay. So okay. So again, we have our distribution. That's what I mean by we need to know this distribution really well. So once again, here's the mean, okay? we said we want 95% coverage or confidence interval. So let's say that ranges from here to here. Okay, so this region here represents 95% confidence interval. That means that this area here and this area here together add up to 5%. Okay, so this area here and this area here together add up to 5%. Or to put it in another way, this is 2.5% and that is 2.5%. Okay. So when we do the statistical test and all of those things, if our data falls in this area, okay, within this 95% range, we say that it is not significant. If it falls outside in this area, okay, so this area or this area, so the 5%, then it becomes significant. So let me go back to this figure here. Okay, so the critical value is 1 minus whatever your confidence interval is. Okay, so if the confidence interval is 95%, okay, 0.95, then the alpha value is 1 minus 0.95, which is 0.05. We often use this number. This is like the golden number in statistics. Okay, we often use this as our cutoff. There are a couple of other terms you need to know as well. Things like one-tailed or two-tailed. Again, you don't need to know this in the context of the HSC exam, but if you're doing a statistical test, you kind of need to know what is one-tail or two-tail. So, it basically goes back to the null hypothesis, okay? The scientific hypothesis and the null hypothesis. So again, it's a little tedious. It depends on whether you're looking at, so if it's a two-tailed, it means that, so let's say you're comparing two groups, okay? A treatment group versus a control group. And let's say if your question is asking, is there a difference between the treatment and control? Okay, you don't care about what type of difference. All you want to know is there a difference between treatment and control? That type of a test is called a two-tailed test. The other type of question you can ask is treatment greater than control? See that here on the right? Is treatment greater than control? Or is treatment less than control? Okay, you're only looking at one side of the graph and therefore they're called one-tailed. The reason this is important is that when you select a t-test, sometimes it'll ask you, is it a one-tail or a two-tail? Okay, all right. It's one of those things that you do just need to be aware of it. Another thing about t-tests is whether the t-test is unpaired or paired. Okay, I know this is a lot of information. I'm sorry about that, but just to give you an overview of some of the terms that are used in these statistical tests. Okay, so what do you mean by paired or unpaired? Another terminology that's used is dependent or independent. Okay, so let's look at this particular example here shown in this figure. So in this particular study, the researchers wanted to know if all of these preterm infants gain weight. Okay, and so in this sort of a study, each child or each baby is measured twice, okay, at the start of the investigation and at the end of the investigation. So each baby has 2 data points, before and after. So this is called a paired t because you get a pair of measurements, okay? The other type is where we compare groups. So here we have, again, a group of preterm infants. One group is given breast milk, the other is given bottled milk. Now, because each baby is, either in the first group or in the second group, each baby is only measured once. This is called an unpaired t-test. Again, this becomes important when you're selecting the t-test. So here's some terminology that you need to just be familiar with it. Okay, you don't need to know all the details about any of these things. That's absolutely not required. You just need to know these terminologies, okay, because they may appear in the exam. Okay, put that all aside. Those are all the technical terminologies. How do we do a statistical test? How do we do an inferential statistical analysis? This flowchart tells us in a nutshell what we have to do. So let's start at the top on the left. So imagine you've got your scientific hypothesis. Imagine you've planned and conducted your investigation. Imagine that you've got your data set in front of you. Okay. Now, let's say you've got two groups, treatment and control, two data sets. And now you're just about to start the statistical test. So first thing you have to do is define the null hypothesis. Remember, the null hypothesis nullifies the scientific hypothesis. So define the null and alternate hypothesis. Okay. If required, okay, not all tests required, but if required, decide whether it's a one-tailed or two-tailed. Okay. Then decide on a confidence interval. So normally I said we take 95%. So if we take 95%, then our alpha value will be 0.05. Then think about which statistical test we will conduct. And I'll come back to that in just a tick. Then conduct the statistical test. The statistical test will tell us how do we compare, will give us the p-value. Okay, compare the p-value with the alpha value. Okay, the alpha, remember, is 0.05. Okay, that's what we normally set it at. So compare whatever the test result says, the p-value with the alpha value. And then you have to make a decision. So if p is less than 0.05, we reject the null hypothesis. So we say that the null hypothesis is not supported, okay? And we will then accept the scientific hypothesis. On the other hand, if p is equal to or greater than 0.05, then you have to accept the null hypothesis. That means that the scientific hypothesis is not supported. Again, don't worry about this. I'll take you through that process in just a tick. And then finally, you're going to report on your results. And make sure that when you're reporting these statistical tests in your results, that you actually include all of the data, the data from the statistical test. Okay. This table is the important bit. Okay. The p-value, remember, the p-value is provided by the statistical test. So if you do a t-test, if you do an f-test or whatever it is, it gives you the p-value. Alpha generally is 0.05. Okay, so if p is less than 0.05, the null hypothesis is rejected. Okay. And if the null hypothesis is rejected, that means that the scientific hypothesis is statistically significant. On the other hand, if p is equal to or greater than the alpha value, then the null hypothesis is true. And if the null hypothesis is true, that means that the scientific hypothesis is not supported. This is the important bit. For all of the statistical tests and so on, this is the important bit. Okay, so this table here. And once again, I've got a flow chart to just summarize that process. Let's start here, scientific hypothesis. So from the scientific hypothesis, we develop the null and the alternate hypothesis, okay? Then we conduct the statistical test. Notice that the statistical test is only conducted on the null hypothesis. It's never conducted on the alternate hypothesis, okay? Really important. The statistical test, once again, is always conducted on the null hypothesis. So you do the statistical test and you get two outcomes, p less than 0.05 or p greater than or equal to 0.05. If it's less than 0.05, what happens? You reject the null hypothesis. And at the same time, okay, you accept the alternate hypothesis, which means you report in your conclusion that the scientific hypothesis is supported. By the same token, if your p is equal to or greater than 0.05, what happens is you have to accept the null hypothesis. If you accept the null hypothesis, you reject the alternate hypothesis. If you reject the alternate hypothesis, you'd say that the scientific hypothesis is not supported. So these flowcharts may be useful for you in terms of analyzing information, statistical information. So I'm not talking about doing the test. I'm talking about what would you do with the data after you've conducted your statistical test. Okay, so all of this will obviously, there's a lot of theory here, there's a lot of information, but it becomes easier when you actually are using, I guess, solving problems. Okay, now I've put in a small section on measurement of error and uncertainty, but I won't go through that today because we don't have the time for it. I want to go back to this figure once again. Okay, so now let me go back up. I'm here right now. I'm talking about statistical tests. So this beige box here. Okay, what do? We go back to the original flow chart. Okay, so remember there's the different types of statistical tests. So what do? Step one, determine the type of data. Whether you've got qualitative or quantitative data. Okay, so that's the first branch, okay, right up at the top, the first branch of this decision tree, of this flow chart. So once you've decided, okay, if you're working with qualitative data, ignore the left-hand side of this flow chart, focus only on the right-hand side, because you're going to be doing a chi-squared test. Okay, so if it's one group, choose the goodness of fit. If it's two groups, it's the contingency. Simple and straightforward. If it's qualitative data, there's only one test that you need to know. Okay, what happens if your data is quantitative? That means you're going to ignore everything on the right. Just look at the left. Okay, so here you've got quantitative. Again, let me zoom in a little. Quantitative. Now look at what are you trying to establish? Are you trying, so once again, going back to our example, think of the two data sets that we got. Are we trying to establish relationship, or are we trying to compare the difference between the data set? If you're trying to establish the relationship, go all the way towards the left and do Pearson correlation. There's only one tool for that. If, on the other hand, you want to measure the difference, so ignore this relationship branch, look at the difference branch. Then the next question is, what data have you collected? Are you going to look at variance data? And if that's the case, you'll do the F test. If, on the other hand, you're going to compare means, then determine whether you've got a single data set. In our example, we've got two data sets, so we'll do a two sample t-test. Okay, so step by step by asking a series of questions about the type of data that we're working with. You can use this flowchart to determine the type of test that you can use. Okay, now, how would this be useful? Let me just go back to these. Sorry, I'm just going to view and navigation pane. And let me look at, and I think it's question 6. No, question 7. So here, this is the question from 2019 exam. Students were given this particular study. Okay, here's a summary of the study. And here's some data associated with it. So here's a table with some measurement data. You're given the means and the standard deviations in brackets. You're given a second data set. Now, look at this question here. Okay. Explain how statistical analysis could be applied to support a decision about which dog substitute is more effective. Okay. In your answer, include a specific test for each data set. So the question is asking, looking at the data, what type of statistical test would you use? Okay, it's a 5-mark question, which means you really need to look at the data, recommend a type of test, and then explain or demonstrate your reasoning. Okay, and so you can then use that flowchart, which I showed you in the previous screen, okay, to look at this data. If you look at this data here, okay, that's quantitative data, okay, because time can be subdivided, it can be fractionated, it's a continuous data set, okay? Again, here, time. What are we doing? We're going to compare these two data sets. We've got two data sets, okay? One group was given a toy dog, the other was given a robotic dog. Okay, so we're comparing two data sets. So if we went back to this, okay, so we're looking at quantitative data. We're looking at the difference. We are given the means, the average, okay, and we're comparing two groups. So this would be a t-test. Okay, let me go back to that. And here in the sample answer, okay, you can see this part here. Table 1 contains two groups. Okay, let me just stop there. Okay, table 1 contains two groups of continuous data with similar standard deviations. The means of the two groups can therefore be compared with a t-test. See, that's what that flowchart tells us as well. But the second data, which is this one here, okay, if you look at this variable here on the x-axis, watch, talk, hold, wave, pat, that's qualitative data, okay? Nominal qualitative data. And remember I said with qualitative data, there's only one test you need to know. And you see that here in the sample answer. Okay, now that's the reason why that information which I put together as a flowchart might be useful. So you don't need to know, sorry, let me rephrase that. You don't actually have to do the statistical test in the exam, but you do need to know what are the different types of tests, what are the conditions in which you'll be using each of those tests, okay, whether you're comparing differences, looking at relationships, whether it's qualitative or quantitative, that sort of thing, okay? So this example tells us about how we can use that data. Okay, so for the rest of this document, I've got a bunch of questions. Here's, and they sort of look at different types of research scenarios. And you can do this test yourself, okay? So for example, here, a biologist is studying the effect of a new plant formulation on the petal lengths of these two iris plants, okay? The biologist believes that this, oh, I've got a typo there, formulation improves the growth of the Virginica species, but not the other species, okay? If you continue further down, you're told alpha value is 0.05, okay, that's the standard. And then you're given these results, okay? You're then told, here's A, sample B. A stands for Virginica and B for Centosa. You're given the means. You're told that a t-test is done, okay? And here we have the p-values, okay? So the p-values here, one tail, two tail, they're identical. So don't worry about whether it's a one tail test or two tail test. Now, this is fairly typical, the typical type of exam question, because you're given the scenario and then you're given the results. And then you're asked, and I've got a whole bunch of questions for you to do, but essentially right at the end, write down a suitable conclusion for this experiment. So again, if you look at the p-value, which is less than 0.0001, that is less than 0.25. If, remember that table that I showed you earlier, which is hypothesis testing. Okay, come back down here. So P is less than alpha, you reject the null hypothesis. Right down here, the scientific hypothesis is statistically significant. So the scientific hypothesis here is supported. Okay, that's what these sorts of questions allow you to practice, your ability to look at a statistical output and then use that as evidence to support your conclusion. So this one.
Sorry.
Yep.
Sorry. I just missed this a second ago. Ian had a couple of questions about paired and unpaired tests. Okay. And so the first one there is, do the students need to distinguish between paired and unpaired tests? And do they need to distinguish between goodness of fit and chi-squared tests?
Short answer, no. You don't need to distinguish between the different types. So they're essentially subtypes of the t-test and subtypes of the chi-squared test. When you're doing this in the classroom, okay, so when you're actually using the data with all the different software, at that point, yes, you will need to make a decision as to whether you're using the chi-squared goodness of fit or chi-squared contingency. But in the exam, you will not be asked to distinguish between them. Okay, that's too complicated. So if you were to just say a chi-squared test can be conducted, that's good enough. But where you do see things like one tail and two tail, it's in the outputs. Sometimes you see this information that's provided. All I'm saying is, don't let that confound you. Don't let that throw you off, okay? When you see something like this, P one-tailed, P two-tailed, and if those numbers turn out to be different, don't get scared. Don't get put off by that. Ultimately, if you're not sure whether it's a one-tail or a two-tail, just pick one of the two. It doesn't matter. If in doubt, pick two-tailed. Okay. If in doubt, pick two-tailed. So And I hope that's answered your question. So again, once again, you don't really need to know the difference when you're answering questions like this. You do need to know that difference when you're using various software to do a chi-square test or a t-test. Okay, I'll get back to this. We've got about 15 minutes now. Question 2 is a similar sort of question, but this one shows, oh, sorry, question 2 is also actually very similar. But it's looking at a different test. It's a Pearson correlation test. Once again, you're given the information and you're given the output, okay? So Pearson's correlation, I don't have time to go through this now, but Pearson's correlation gives you what's called the R value and the R squared value, so the correlation coefficient, but right at the bottom, you also get a P. So all of these can be used, again, to check whether the hypothesis is supported or not. Question 3, if you look at the data, okay, look at the variables here. Okay, that's qualitative. Okay, in fact, both types, both variable qualitative. So you would use the chi-square test. Chi-square test also gives you the p-value. Compare that to the alpha value. And once again, it's that same process. What I'm trying to explain to you here is get into that mindset. Okay, all we're doing is comparing p with alpha. That's what all of these inferential stats are trying to do. So whether it's the Pearson correlation, F-test, t-test, chi-square test, it doesn't matter which test, we're trying to compare p with alpha to look at the level of significance. And just to make things a little bit more interesting, I've given you 2 other questions for you to just try on your own. The main thing about this, okay, is that the data sets are actually in the Excel spreadsheet. Okay, so that, let me go back to that wet question again. So this is the question, this is question 4 on, which is looking at antibiotic use data, okay? The question itself is relatively straightforward. It's exactly like the others, but just so that you get a bit of practice in doing this, I've deliberately chosen a large data set. So here's the Excel spreadsheet, okay, and it's the worksheet labeled antibiotic resistance. Here's the description of the study, and here are 3 columns of data, okay. Now, the data, so there's the raw data for you to play with if you want to. And then there is a set of questions for you to answer, again, using the statistical data or the statistical output to support your answer. Now, relationship is a Pearson correlation coefficient. Okay, so that means select all of this data into Excel, or you can actually use this Excel spreadsheet, and you can then do a Pearson correlation coefficient. Okay, so right up here, and you can go, I think, so that's where the Pearson function comes up. So you then select the 2 columns, the 2 arrays here. Okay, so array 1, if I minimize it, I'll just go array 1, and then array 2, which will be here to, oh, don't know what happened to that row, but anyway, if you go, okay, yeah, the feeling that, oh, let me go back to the Excel, sorry. Going back, starting that again. Array 1 would be I'm going to leave the bottom row out. And then array 2 would be from there to. And that then will give you, I haven't got it here. I won't diagnose it now, but essentially, when you do the Pearson correlation, it will tell you what the correlation coefficient is, okay, 0.82. So you can then do that analysis here for this question, okay, and then answer that particular those two questions there. The next one is, this is a Donald Trump question inspired by him. So you know that there was a time when there was this push that to use hydroxychloroquine as a potential treatment for COVID-19. So here's some actual data. So I've got it as a separate spreadsheet. Now this is a huge data set. So I just wanted you to get the hang of working with large data sets. Okay, so you can see just over here, it's got 820 odd rows of data. So you can just get the hang of working with large data, because that's one of the things that the syllabus would like you to do, to have that familiarity of dealing with large data sets. So that's an example here. Okay, so again, go through and what recommendations would you have to make? The questions 6 to 8 are all from past exams or the NESA sample exam. I want to just spend a little bit of time, about no more than 5 minutes, on this particular question, because it kind of highlights the depth of reasoning that's expected. Okay, so this was the NESA sample exam, which was released in 2018. And the question goes something like this. I'll just give you 30 seconds to read that top part here. Okay, so we're told that they want to use this concrete formulation. The minimum strength is 22 megapascals. So they've got two treatments. Okay, one set was given treatment 1, the other set treatment 2. And then some strength data was collected. You're then given some information about the statistical test that was performed. You're told that it's a t-test. You're told that the p-value significance level, the alpha value, is 0.0001. Okay. Now, here's the data. So what the question's asking, what conclusions can be reached about the relative strengths of the two types of treated concrete and the suitability for the intended purposes? Justify your answer. Now, the first thing we can look at is, look at the p-value here. So the p, and here you see the word two-tailed has appeared. Don't worry if you're not sure about what the two-tail means. So P has a value of 4 × 10 to the minus 27. Now that's much smaller than this value here. So the calculated p value is smaller than the alpha value. Okay, let's look at the sample answer. And I want to, and the reason why I want to focus on this is to tell you the reasoning. So when the p value is so small, and it's so much more smaller than the alpha value, okay, look at the sentence here. P less than 0.01 means that the scientists can reject the null hypothesis and accept the alternative hypothesis. Therefore, that the difference between the two treatments is significant, statistically significant. Make sure you use those terms in your answers. If you have questions along these lines, we are asked to justify the conclusion. Use words like statistically significant, because that shows that you have the command of the vocabulary, okay? You're comparing it to the p-value. Now, let's continue. However, the t-test on its own cannot be used to determine whether the treatments are fit for intended purpose, okay? Look at what they've done here. They've taken the mean, okay, and subtracted 3 times the standard deviations, okay? So what they're doing here is now they're using the descriptive statistical data there. You've got in that table the mean and the standard deviation, okay? Remember I said in the standard, for the normal distribution, one standard deviation is about 68% of the data. 2 standard deviations are 95% of the data, and 3 standard deviations are 99.7% of the data. So what they've done is they've taken the standard deviation, 0.31 or 0.28, okay, and multiplied it by 3, okay? So that's what they've done here, okay? 0.28 times 3, so 3 standard deviations. They subtracted the mean, okay? The mean, which is here, 23.4, okay? Minus 3 standard deviations, which then gives us this value, okay? 22.56. They go back to your original question up here, okay? The minimum requirement is 22 megapascals. So the mean minus 3 standard deviations is still larger than the 22 as the minimum, okay? So that means that 99.7% of the samples that were tested are stronger than the minimum requirement. Notice how all of that information has been woven into this answer, okay? So since 22.56 is well above the minimum strength, the scientists can be confident that treatment 2 satisfies the minimum strength requirement. And then they go on to summarize the findings. In summary, the scientists checked that they could use the t-test with their data and then carried out the test. And then the analysis showed that there's a statistically significant difference with treatment 2 having the higher mean. Look at the wordings here, okay? The mean for treatment 1 is below the minimum, so it is not fit for the intended purpose. The mean for for treatment 2 is higher than 22 megapascals. And the scientists then established that almost all of the concrete treated with treatment 2 would have a strength higher than 22 megapascals. The scientists can recommend the use of treatment 2 for strengthening the concrete for the intended purpose. That's a very high quality response because it's not, and you know, students don't have to do any calculations other than this one simple calculation here, looking at the standard deviation. Everything is based on the stimulus information provided here, okay, and the information from the statistical test provided here. Just using that, analyzing, taking your time, okay, to construct that response is really, really important. Ensure your reasoning. Use proper terminology. Those are the things that the examiners are looking for. It's not so much, about the mistakes that you might make in terms of, which numbers you're using or something like that. Okay, it's in your reasoning. The reasoning is the most important thing. That's what they're expecting for science extension students. Remember that this course is not about specific concepts in biology or chemistry or physics or anything like that. This is about scientific understanding. And in module 3, it's about what you do with data. Okay, so and then the other 2 questions are generally straightforward. I've got sample answers there. And again, you can see the same reasoning coming through. So I'll stop the presentation there. And Bill, are there any other questions? If not, I'll hand it back to you.
No, I think all the questions are there, unless anyone is going to pop something else in the chat box there. Thanks a lot for that, Sean.
No problems.
Yeah, it's I think one of the main.
Thanks, Sean.
Yeah, I think the main problem more than anything else is that the statistics side of it, it is kind of complex, but it's not, and it's something that it's something completely different to what we're used to doing in a lot of our courses. So, thank you very much for that.